Hey friends, I haven't been teaching for about 3 weeks now, and I'm beginning to feel the itch. It's particularly itchy because it's been even longer since I taught Evo-Devo. I've mainly been focused on cell biology and genetics, since we don't even offer a developmental biology course here. I've occasionally taught evolutionary biology, and a few times I've taught an Evo-Devo course. And last year I taught a history philosophy course on evolutionary thought. But it's been a long time since I had an opportunity to bring all those concepts together and fuse them into a messy souffle of convergent ideas. So today I'm going to be lecturing on the history of developmental and evolutionary ideas. You're going to hear some very old stuff and some relatively new stuff, but they're all going to fit together, I hope. I also intend to reveal that the roots of our understanding of mechanisms of biological change are more than 200 years old, and that creationists are even more out of date than is commonly believed. The evidence against them has been piling up for a long, long time. So, I'm beginning way back in the era before Darwin, when the natural historians of the 18th and 19th centuries were starting to integrate embryology and anatomy and coming up with wild theories that would coalesce eventually in Darwin's descent with modification. Initially, though, their hypotheses and observations were entirely compatible with some modern versions of creationism. I'm actually a little surprised that creationists nowadays aren't big fans of Cuvier, because he was vehemently opposed to any claim that organisms of one species could change in any significant way into another species or even have any substantial variance. Cuvier is considered the father of paleontology. He was also an expert on comparative anatomy. He was the kind of expert that if you dug up an old tooth or a broken femur, you could send it into Paris and he'd be able to identify the animal precisely and reconstruct the rest of the animal and tell you about its diet and behavior and so forth. He based this determination on his principle of the correlation of parts. The idea that every single piece of an organism is so perfectly integrated into the whole that no aspect could be changed without making the animal deformed and unable to survive. This was the basis of his objection to evolution. It ought to be familiar to creationists. It's pretty much the same concept as irreducible complexity, only it's over 200 years old. Michael Behe has never had an original idea. However, Cuvier also had held views that might have made modern creationists uncomfortable. He knew that geology revealed a complex history of the Earth. He was a catastrophist who believed that major events had caused massive extinctions. He didn't think there was just one. The biblical flood was not enough. He thought there had been multiple succeeding catastrophes that required multiple creation events to repopulate the Earth. He could look at the geological record and see evidence of faunal succession, that there were previous eras with very different organisms living in them. He thought there was an earlier age in which reptiles rather than mammals had been dominant, and he was able to identify large fossils as being reptiles. He died before Richard Owen coined the name dinosaur, but he would have been perfectly fine with the addition of a whole new class to the systematics. He expected that there were all kinds of novel animal forms in the past, and in different parts of the world. His God was a creative agent who was always coming up with new species. There were limits, however. For some mysterious reason unknown to the mind of man, animals could only exist within 4 branches of classification: the mollusca, the radiata, the articulata, and the vertebrata. The body plans of those 4 types are incompatible, and no transformation from one to the other was allowed. Of course, he didn't allow any gradual transformations within a branch either. He had critics. One you have all probably heard of, Lamarck. Lamarck's objections were relatively mild. He accepted a limited amount of gradual variation within a species, arguing that animals could modify their forms through use and disuse, but he still agreed that the main branches were inviolate, and that there was no way a mollusk could transform into a fish. In my opinion, Lamarck agreed with Cuvier on all the essential points. Lamarck just believed that the body plan had more slack and was more responsive to the environment. Cuvier had a more radical opponent in Geoffroy St. Hilaire. Geoffroy wanted to tear down the whole structure of the four branches of Cuvier. He was a believer in the unity of organic composition and thought that there was one principle of animal form and that everything from lobsters to flatfish to mice and to octopuses were built on one plan with variations. This is a view that conforms to modern ideas of common descent with modification. But, make no mistake, Geoffroy was a deist who believed in the Enlightenment idea that the world was a product of natural law, that we had a watchmaker God who set the pattern in motion, and in his case even thought that this God created individual species. So he was no evolutionist. He thought in terms of archetypes. He thought that God had blueprints that he used to create life following certain ideal patterns. These forms of life would then respond to their environments to modify those core patterns to create the species we see today. But he didn't have a model of descent leading to heritable diversity. He had some other wild ideas based on observations that are now familiar. For example, he proposed the concept of unity of composition, that everything was built of the same parts, rearranged, and resculpted to fill different roles. We are all familiar with illustrations of homology, showing that a human arm has the same bones as a bird's wing, where one can see a one-to-one correspondence. That's evidence that they were built on a common plan with modifications, and even if you disagree on the mechanisms behind those similarities, Geoffroy made the same argument, but then extrapolated it even further, to a point that now looks kind of ridiculous. He compared the structure of a fish to a lobster, and he was drawing connections between the bones of one to the hard parts of the other, and concluded that a lobster was a modified vertebra, that the invertebrate was structured inside of a vertebra rather than wrapped around the bony spine. He correlated the bumps and spiny protrusions of the vertebra to the limbs and exoskeletal details of the crustacean, and argued that a lobster was a fish lying on its side in order to make the correspondences work. So in 1830, Geoffroy and Cuvier had a prolonged debate on this topic, and Cuvier routed him. The idea of a unity of form seems to have appealed to the audience, so it wasn't a career-ending performance, but still, he had chosen to build his case on a correspondence that was rather ludicrous, and that has most definitely not stood the test of time. What was lasting, though, was a hypothesis made by the way. If a crustacean was a fish on its side, then organs inside this imagined vertebra would have rotated 90 degrees to restore a dorsal-ventral axis. And by chance, it ended up at the 180-degree rotation relative to the fish. The body of a vertebrate would have been upside down relative to an invertebrate. Geoffroy also emphasized the role of the environment in shaping the organism, and that has also stood up well, becoming even more important with Charles Darwin's revelations. But yeah, I have to appreciate that the radical morphological transformations that he argued for look silly. A lobster is a fish lying on its side. How absurd. How could the transformation have occurred? A fish just laid down and sprouted legs from the dorsal and ventral fin rays. It is not surprising that he lost the debate to Cuvier, since he didn't have a plausible mechanism to cause that change. And yet, there were other observations that suggested that there was a radical inversion in the distant past. Fish are deuterostomes. In early development, during gastrulation, the closure of the blastopore is the site of formation of the anus. Lobsters are protostomes. The closure of their blastopore leads to the formation of their mouth. The early embryos of the two do seem to be inverted versions of each other. So maybe there was some weird flip that was executed in the creation of the two, as if God were playing with the same core body plan, generating novel versions by turning modules upside down to create some new permutations. Alternatively, as Geoffroy was implying, there could have been a natural process that would allow organisms to make a dramatic metamorphosis between forms with astonishingly different results. This is what evolutionary biologists expect happened, but not in the abrupt way Geoffroy envisioned. There was a common ancestor that spawned two lineages, one that adapted the ancestral body plan to function in one way, and the other that adapted with the reverse pattern. Geoffroy's argument seemed silly at the time because he didn't have access to modern molecular biology. He was only looking at bones. The idea that vertebrates and invertebrates were inverted relative to each other was confirmed by looking at genes in the 1990s. The same molecular signaling pathway establishes the dorsal-ventral axis in protostomes and deuterostomes. So the last common ancestor had this same pathway, but one lineage used it in an inverted fashion relative to the other. This pathway is the cordon morphogenetic pathway, and it uses diffusible signaling molecules to generate a gradient defining the top and bottom of the animal. One side of the animal is set up by a gradient of a molecule called bone morphogenetic protein, BMP, or decapentaplegic, DPP. BMP, DPP have different names in vertebrates and invertebrates, but they're the same thing. DP was found and named in flies, while BMP was found and named in vertebrates. The names are both so well locked into the literature that there is no point in trying to reconcile them. You don't want to rename these commonly used terms. So we're going to have to live with the dual naming of a common molecule. In vertebrates, BMP, DPP are localized to the ventral portions of the animal, where they are secreted, and they diffuse dorsally. In flies, BMP, DPP is located dorsally, along the back of the animal. On the other side of the animal, cells produce a molecule called cordon, or short gastrulation, SOG for short. These are also homologs, with cordon as the vertebrate molecule and SOG the invertebrate version. Cordon SOG is localized to the dorsal side of vertebrate embryos and to the ventral side of invertebrate embryos. Let me tell you, it can get confusing teaching this stuff with two different names for each of two signaling factors that are expressed in different reversed orientations in two major clades of animals. It's what you got to know, though, if you want to read the scientific literature. The embryo contains a diffuse low level of BMP-DPP expression. It's kind of the default field. Cordon SOG is secreted in large quantities by the gastrulating tissue, and its role is to bind to BMP-DPP, inactivating it. In vertebrate, it's produced by the dorsal lip and produces a streak or a line of cells to make more cordon SOG. which in turn is transported by facilitated diffusion or flux to the ventral sides of the embryo. But BMP-DPP also activates toloid expression, another molecule shared by vertebrates and invertebrates that cleaves cordon and releases BMP-DPP. I know it doesn't sound like it, but the idea is simple and was proposed by people like Alan Turing in the 1940s. Setting up a pair of complementary gradients in an organism is an elegant way for cells to calculate their location. This is biology, though, so it quickly becomes increasingly complex with multiple supporting pathways and multiple interactions that cells use to read the gradient. But that's the way biology is. It's a process constructed by incremental tinkering over millions of years. It works, but it's not simple. This is a dynamic system. Cordon-SOG sets up a self-organizing morphogen gradient, inactivating and opposed by BMP-DPP, to basically set up a standing wave of varying concentrations of signal molecules from dorsal to ventral. It's beautifully balanced, and it's the same gradient in fish and lobsters, just inverted relative to each other. We can safely assume that the last common ancestor of all animals evolved this mechanism of generating a gradient of positional information that has persisted for at least 600 million years, and possibly much longer. But we still have a little problem. What kind of ancestral form could flip its descendants from one orientation to being upside down and still retain functionality? That still seems like a silly idea, at least if you're imagining modern large adult animals and picturing them upside down. You wouldn't do that, though. You know we have to compare embryonic forms as a model for pre-Cambrian ancestors. When you do that, the transformation becomes much more readily feasible. We quickly find examples of organisms that straddle that line between upside down and right side up. So this is the trochophore larva of a modern protostome, an annelid or a mollusk. It has a mouth, a gut, and an anus. It also has an eye spot and a ciliary tuft, that is an apical sensory organ. The mouth is surrounded by ciliary bands that beat and propel tasty bacteria to the mouth. So this is a feeding structure. On the other hand, this is a deuterostome larva of a hemichordate or, for instance, a sea cucumber. The mouth, gut, anus, and apical tuft, eye spot, and ciliary bands are all there. It is very similar to its cousin protostomes. These larvae are much closer in appearance to the last common ancestor of protostomes and deuterostomes, and it's much easier to evaluate how an inversion could occur. This pattern emerged deep in the pre-Cambrian among free-swimming planktonic organisms. It was adaptive for both harvesting marine bacteria and for dispersal. The next step in their evolution was to grow larger, sink to the bottom where algal mats formed a much richer food source, and only then committing to one side or the other, gnawing, feeding, walking, all those functions. And the difference in the two sides was much less dramatic then. All the hard work of forming the tissues of the adult were already done and set in place, and their descendants would form the specializations we now see. I think one of the points here is that I'm often confronted by creationists who insist that it is impossible for organisms to evolve. They have only the most shallow knowledge of biology and claim, for instance, that cats can never turn into dogs because those are the only animals they know. And they think the process is about transforming an extant animal into a different extant animal Further, they think morphological change is somehow forbidden. If you know the science, though, you're aware of all the evidence telling us that life on Earth has manifestly changed over millions of years. And we know that by looking at their constituent molecules, that they have shared genes that clearly demonstrate their relationships. These are not topics for debate. It's settled science. It's fact. Personally, I think everyone ought to know more developmental biology. I was raising insects in my bedroom as a kid, watching embryos turn into larvae and becoming adults. That was a great hobby, although at that time I raised mosquitoes in a jar in my bedroom. Turned out to have some downsides to it. In college, I studied fruit fly metamorphosis. In grad school, I studied zebrafish development, observing eggs becoming fish in a day. And as a postdoc, I watched the neurons of the developing grasshopper nervous system form local networks. Everything is about change. It's all change. Nowadays, I have to give the side eye to those ignorant people who deny change, even as they are having children and aging themselves. Every time I dive into a biology journal, I see nothing but documentation of how the world, the environment, the life on this planet are engaging in the process of transformation. Embrace it and enjoy it. Be intellectually challenged by it. This is the part where I give credit to the people behind the scenes. See all those names scrolling by? Those are the people who feed my spiders. Not in a grisly way, of course. They contribute to my Patreon account at patreon.com/pzmyers. You could feed the spiders too, just by going to patreon.com/pzmyers. Everyone who's watching this video is also contributing. You just don't get credit, I'm sorry to say. But thanks. The little revenue I get from these videos is going into maintaining my lab and my spider colony, and also, of course, to keeping free thought blogs afloat. That's right, I'm also part of the blog network at freethoughtblogs.com that includes a lot of other interesting writers. You should check them out. And what you're seeing is just my usual walk into my university, the University of Minnesota Morris. It's a pretty place in the Midwestern prairie, although there sure are an awful lot of trees for a prairie.